You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts, and I have Dr. Ilan Morad. He's the founder and CEO of Evolution Biotechnologies Ltd. So, Dr. Murad or Ilan, thanks for coming.
Hey.
I appreciate you being here.
Thank you.
And you're over in Israel, or where are you?
I'm in Israel.
Okay, what time is it there right now?
Now it's 8 p.m.
Well, thank you for doing this so late. I appreciate it.
Thank you.
So tell me about Evolution Biotechnologies. What does the company do?
Okay, the company was established 19 years ago in an incubator in Israel, in Weizmann Science Park, in the Weizmann Institution. And the basis of the company was a technology that we now use, and we have patents all over the world. It's a screening platform. And the outcome of the technology are therapeutic peptides. So actually, we can use this platform to discover novel therapeutic peptides to almost anything we want. We did several projects for therapeutic peptides in collaboration with other companies. And after a while, we decided that we want to do our own projects, and we decided to focus on cancer. So at the beginning, we wanted to do what everyone else is doing, and just take the same targets as companies used to generate antibodies to, and to do the same with peptides. And actually, we succeeded doing that, but then we decided that this is not what we want, because we would have the same, more of the same. And the same is drugs that give relief for a limited time, and then the cancer returns. So we decided that we want to make an effort and try to think how we can cure cancer. Because we have such a great tool that we can generate therapeutic peptides, and it's our own platform, and we can do great things with it. So
well, let
me try to do something.
No, that's far better to cure cancer than to just have drugs that keep it quiet just for a few months. But can I ask you? Can you explain some basic things for people that don't know what are peptides and how, where do they come from in the body? What are they used for?
Yeah. So peptides, the simplest way to describe peptides is microproteins. So if a protein is a large molecule, so if, for instance, the size of a protein could be something around 50 kilodaltons, and peptide could be 1 kilodalton, 2 kilodaltons. So they're much smaller, but they are consist of the same building blocks, amino acids. So protein, probably everybody knows what proteins are. So peptides are very, very small proteins. You could say that.
And where are peptides produced in the body?
Oh, there are peptides in our body. Some of them have biological activities. Some of them are pieces of proteins, are broken proteins. And they have the role in our physiology, but our peptides are completely synthetic.
Right.
So what we do in our platform technology, we just make what we call libraries of peptides or large numbers of peptides, usually millions or even billions different peptides. So among this huge number of peptides, we can pick up peptides that do anything we want. We just have to know how to isolate them, and this is what our technology does. So
one question: what kind of roles do peptides have in the human body? What kind of things do they do?
Oh, sometimes they are hormones. For instance, the insulin is a peptide.
Oh.
But insulin is a big peptide. Our peptides are much smaller than insulin. They say that the definition of peptide is up to 100 amino acids, but this is actually, you can say, a small protein, not a microprotein. Our peptides are very small, something like 10, 12 amino acids. So they're very, very small.
Do peptides fold like proteins do, or do they stay in a fixed structure?
Actually, they don't have a defined structure. So this is, big protein has a very solid structure, and sometimes this is one of the problems. For instance, antibodies are proteins, and a very large protein. For instance, the size of an antibody is 150 kilodaltons, while the peptide could be 1. So there's a great difference in the size, and this is good for the peptide because if you want to manufacture protein, you have to do it in cell cultures and use special plants for that, and this is very complicated and expensive. And peptide, you just click the sequence that you want on a machine, and you get the peptide on the other side exactly what you wanted to. So they're much cheaper, easy to produce, and they have other qualities. So that's why we decided to use our platform technology to generate peptides, because we also could have do it with antibodies, but we decided that we want to focus on peptides. We think it's better. So at first we wanted to discover cancer as everyone else is doing, and then we decided that we want to do something else. So first we thought a lot about what are the main problems with cancer, why cancer always comes back, what is called a drug resistance.
Mm.
And another question that we face is, why are there so many side effects? Chemotherapy has a lot of side effects, and this is well known, but even with very specific antibodies, there are some side effects that could be not very simple. So we ask ourselves, how come they are so specific? How come there's side effects? So after a lot of thinking, we understand what the problems are. So the main problem with cancer is that the cancer cells are very mutagenic. So they can generate mutations that can help them escape almost any therapy, and this is exactly what happens.
Right.
So when you give a good drug to the patient, many times it seems like it helps, but then what happens? Not all the cancer cells are destroyed, and then some of them acquire a mutation that disables the treatment. And then they start growing more and more, and then you get drug resistance, because these cells have a mutation that disables the treatment. So what can we do about it? Now, we had a lot of experiments and a lot of thinking about it, and then we remember something from the 80s, of the last century. You remember AIDS, what happened with AIDS there? People who got AIDS, that was a death sentence for them.
Right.
And then they started generating all kinds of drugs. And then the patient got the drug, it seems to help for a little while, and then the virus, the HIV mutated, and then the treatment didn't work anymore. And then they found the cocktails. And from this moment and on, actually people are not sick if they keep taking the cocktail. We have experience of people who take the cocktail for more than 20 years.
Wow.
Actually, they're not sick. They are HIV carriers, but they're not sick. Remember Magic Johnson?
Yeah.
He's well, he's functioning
Huh.
his HIV carrier, but he's fine, he's not sick. So I remember at the time when it happened, I said, wow, this was a genius thing. Only you have, instead of giving the patient one drug at a time, you give them 3, and then you solve the problem. It's so simple. So now I remember that and say, hey, this is the same problem because, HIV is very mutagenic, and actually most of the viruses are very mutagenic, and the cancer is very mutagenic. So it's very similar. So if they solve the problem by giving a cocktail of several drugs, why don't we do the same here with cancer? It could do the same.
Right.
So actually, all the industry and academia, they go in this direction because, there's what is called combination therapy. They start giving more than one drug at a time. For instance, they give an antibody together with chemo or all kinds of this combination. Usually they reach 2. So we have to go even further than that. I think that at least 3, because, the AIDS cocktail usually is a combination of 3.
But why do you think that a cocktail is able to kill a virus or, stop cancer? Is it because that, these, viruses or cancer can only mutate at a certain rate, and this overwhelms them? It attacks them on so many fronts that they don't have time to mutate to avoid and to be able to survive this stuff?
It's not a question of time. It's a question of statistics. For a cancer cell to get one mutation that would help the cell overcome the treatment, there's a specific probability. Now, if you have to do at the same time 2 mutations, now you have to multiply the probabilities. So the probability of having a simultaneous 2 mutation is much, much lower. Now think about 3. It almost never happened because the probability is so low. And this is the whole story, statistics. The combination of 3 mutations that must happen simultaneously is almost 0. And this is what happened with AIDS, and this is what would happen with cancer. Now, but there's another thing. Now, if you give a cocktail like in the AIDS, a cocktail of 3 drugs, the number of side effects would be tripled. When you give 1, you get several side effects. When you give 2, you get the sum of the side effects. When you get 3, you get more. So it didn't seem so good, but then we had an idea, and this was actually exactly what we wanted to. And everyone who is a biologist knows what is called Avidity Effect. Now, Avidity Effect is exactly the opposite of what I just mentioned. If you have several drugs, let's say, if you give them separately, you get a specific efficacy. If you attach them together and express them in one molecule, you get an exponential growth in the efficacy. And there's been many experiments about it. So, for instance, if you take, let's say there's an experiment that there was an article about. They took one antibody, they got a specific efficacy. They combined it with another one, they got sometimes 100 better efficacy, sometimes even 1,000 times better. So this effect is exponential. So if you give 1, if you go from 1 to 2, you get much better exponentially. If you go to 3, you get even much, much better. So
I have a quick question here. Do we observe that certain cancers mutate along predictable pathways? Let's say you have a certain kind of cancer and you expose it to a known chemotherapy. Does it tend to mutate into known, do we know how it's going to mutate, or can it mutate in completely different ways where you don't know what it'll become?
Yeah, we know it. There are some kinds of cancer that we used to give a specific drug, and they saw that some of the patients do not respond. And then they found that there's a specific mutation that if this mutation pre-existed in these cancer patients, then they didn't respond to this drug. So from this point on, they don't give this drug unless they give a test to see if this patient has this mutation. And if they do, they don't give the drug to this patient because it would be useless. Now, this kind of mutation could happen later on. For instance, they start giving the drug to the patient, it works, and then the same mutation is acquired by the cancer cells, and then the drug stops working.
Yeah, but do these drugs cause predictable mutations where we know if we give a certain cancer this chemotherapy, it's going to mutate in these ways, or is there still room for it to do things we don't see?
Yeah, these things are known.
So have we tried to? Has anyone tried to steer cancer, where you deliberately give it a certain drug because you know it will make it mutate in a certain way, and then you can attack it with a cocktail? Have we thought about doing that, like lure it into a trap by making it mutate?
Yeah, but we're not. But we're not talking about that. What I'm saying is actually personalized medicine.
Okay.
So each patient would be tested to see what is the profile of his cancer. Which actually what we want to test is which proteins that are overexpressed on the surface of the cancer cell and what we call receptors. Which receptors are overexpressed? Means that they are expressed in high numbers. So which proteins or receptors are overexpressed in the specific cancer of each patient? Now, when we know that, we can design a molecule that would have targeting peptides towards these receptors. So actually, by this test, we would know exactly which drug and which combination of targeting peptides to use. Now, each patient would get a different drug, but all of them would look alike. Now, I can tell you about the drug that we invented, which we call the Mutate. So, Mutate is the drug that would cure cancer. And how it is built, it contains a flexible scaffold, like an octopus. Now, octopus has 8 arms. Our octopus, or the Mutate, contains much more, dozens of arms. And on the tip of each arm, there's a small peptide. And these peptides are the peptides that we discover using our platform technology. Now, there are 2 kinds of peptides. The first one are targeting peptides. And these peptides are like an antibodies. They can find a specific cancer target and interact with it. And by this interaction, they do 2 things. First, they inhibit this protein. And second, when this interaction occurs, the receptor grabs the whole molecule and takes it into the cell. And this process is called internalization or endocytosis. Actually, the cell is swallows. The cancer cell thinks it is getting something good, like food, so it swallows it in. So the whole molecule that contains many peptides gets into the cell. Now, here we make a surprise for these cancer cells because the second type of peptides there are toxic peptides. So these toxic peptides are not toxic outside the cell because their target is inside the cell. So outside, they're inert. They do nothing. But when the cancer cell swallows this molecule, then the toxic peptides can find their target, and actually it's a poison pill. So they poison the cell from within. So what happens, again, the targeting peptides on the mutator interact with different target receptors. Then the whole mutator is swallowed by the cell, and then the toxic peptide poison the cell and kill it very efficiently. So this is a combination of 2 strategies. First, the multi-targeting won't let the cancer cells escape the therapy because of what I mentioned before, lighting the AIDS, because the probability of the cancer cells adopting triple mutations simultaneously is actually almost 0. And then there are 2 mechanisms by which the cancer cells are inhibited and finally destroyed. First, the targeting peptide inhibits the cancer from the outside, and then when the whole mutator gets into the cell, then the toxic peptide poisons the cell and kills the cell from within. So these are a combination of several strategies. Now, there's another thing that is good about this mutator. The scaffold is very flexible, again, like an octopus. So it can get into very difficult places or narrow places. For instance, if there's a solid tumor or a large protein like an antibody has difficulties getting in deep into the tumor, our molecule would not have such difficulties because it's very sneaky, like an octopus. Now, another thing is sometimes you give a drug to a patient, and after a while, our own immune system recognizes that drug and starts generating antibodies against it. Then it doesn't help anymore because it is cleared out of our system by our own immune system. Now, the requirements of our immune system in order to recognize these molecules are size. The bigger the molecule, the easier it is for the immune system to recognize it, and to be with a defined solid structure. And these two are not existing in the mutator because it's flexible, doesn't have a solid, defined solid structure, and all the components are very small. For instance, the peptides are very small, so usually our immune system do not generate antibodies against them. And also the scaffold itself, like the octopus, is from materials that do not generate any more response. So this is good. Now, now the second thing is side effects. So as I mentioned, because of this avidity effect, this molecule, the more targeting peptides we would have, the more effective would be the molecule. So we would be able to lower the dose that we give to the patient. And when we lower the dose, it interacts less in other cells that are not cancerous and have the same target. For instance, the targeted therapy goes to specific targets. So in the cancer case, usually they go to cancer targets that are overexpressed in cancer cells. But the same targets exist in non-cancer cells in smaller numbers. So what happens when you give, for instance, an antibody that goes to the cancer cells? It goes mostly to the cancer cells, but it goes also to non-cancer cells all over our body. And this gives side effects. So if we can lower the concentration that we give, then we would lower the interaction with non-cancer cells, and this would lower the side effects. So, and this is all because of the avidity effect. And the avidity effect is caused because we connected all the components of the drug. So the same drug, because it contains multi-targeting, on the one hand, won't let the cancer develop drug resistance, and on the other hand, would lower the side effects. So this is exactly what we wanted. So we made a few experiments to show this principle, and we showed it. We showed that when we give one targeting peptide, we get specific. We have to use specific dose in order to kill the cancer cells. But if we give two in combination, we can kill the cells in much lower concentration. So this is exactly what I was talking about. This is the avidity effect that shows that we can lower the side effects. And of course, the whole thing together with the toxic peptide would be very, very efficient. Now, again, I have to emphasize that each cancer patient would get a different mutator, but all of these mutations would look the same. They would all have flexible scaffold with many arms and with many peptides, some of them toxic peptides, some of them targeting peptides, but the targeting peptides would be different from one patient to another. So
why? This makes sense. But why don't the peptides trigger an immune response, especially if they're attached to a larger molecule? What is it about peptides that don't trigger the immune response?
Because they're very small. So, small molecules do not generate an immune response. Let's say aspirin, okay, or ibuprofen, or Tylenol. They are small molecules. Have you ever heard of someone who got an immune response to these molecules? No, because, you can take it over and over again, over and over for all of your life, and almost all the people won't develop an immune response to these molecules because they're small.
Well, what about a virus? What about a virus? How big are they compared to peptides?
Viruses are huge.
Okay, I didn't know that.
Viruses are huge. That's why our immune system generates antibodies against viruses. But sometimes they can. Sometimes they do their work so fast that before it takes time until our body develops an immune response. So before our body is prepared, it can go into the cells, and then our immune system can't see them in the cells.
So viruses are as big as a lot of proteins, or bigger? Like, what's the scale of size?
Much. Viruses are comprised of many proteins. So
okay.
If I say that, for instance, an antibody is 150 kilodaltons, viruses are millions or dozens of millions in size. So
okay.
they're huge.
Okay, interesting. Yeah, I didn't know that. That's very cool. So have you found the size limit below which peptides seem to be invisible to the immune system?
Yeah, size is important in order to induce an immune response. Yeah, everyone knows that. Now, I can't guarantee that there will be no immune response whatsoever in every patient, because even the small molecules like aspirin sometimes generate an immune response, but it's rare. So I can't guarantee it, but we think that this would be the case based on evidence of other experiments and scientific data.
Okay.
But this is not the important thing. The most important thing in this concept, the Mutate concept, is the multi-targeting together with a toxin and the Avidity Effect. It comes together with this connecting all of these peptides together. So this is the important thing about it. Again, multi-targeting together with the toxin and the Avidity Effect. That's what make this mutator so special, and we're sure that it's going to cure cancer patients, because again, the cancer, like in the AIDS case, won't be able to fight back or to become drug resistant.
Is there any way to make this non-personalized, where it will work for everyone that has a certain type of cancer, or at least large cohorts, or does it have to be completely individually personalized?
I think that the personalized strategy is the best. Of course, we can give it more in general. The first mutator that we're working on is contains targeting peptides towards the most common cancer targets. So probably there will be many cancer patients that would have this combination. So this would be lung cancer, colon cancer, and head and neck cancer patients. I'm sure that there will be many of them that would be fitted to get this first mutator. And we're working on more and more targeting peptides. We think that if we have 100 targeting peptides, then combination of these 100 would cover all cancers. So we're working on it. And I guess that in a few years we get mutators to all kinds of cancers.
Okay,
now,
so
again, we can give not perfectly match mutators to patients, but I don't like it, because the more personalized personalized drug, I think it's much better for the patient, because it would be much more precise, much more efficient, and with less side effects.
Well, it's true, but you have the trade-off of cost. So, I mean, the therapy like this, what if it costs $500,000 to do because it's so personalized, when you may be able to make a drug that might be $1,000 for a condition?
I don't know about the cost, because, we want to be the ones to determine the cost. We are just a small company. that we would have to collaborate with big pharmas, and they will take it further and develop the drug until they have an approval from FDA to sell it in the market.
I got you.
I think start treating cancer patients, we can do it within the clinical trials. So when people would start understanding that this is a good treatment, they are already trying to get into the clinical trials. But we say that we're not there yet, at least for another year. But when we are in the clinical trials, people would try to join the clinical trials, and then they would get the treatment for free. So I think this is where we would start treating people. And about the cost, I don't know. I think that they would have to change the thinking about it. I mean, the FDA.
At what stage? Yeah, no, I know. At what stage will you hand this over for clinical trials, or will you be doing, will your company be doing the clinical trial, or will it partner with a larger one to do it?
Yeah, we think that with the right budget, we can be in the clinical trials a little bit more than a year from now. Actually, we have the first mutator, so what we have to do is just to, the preclinical trials and, of course, the clinical trials are actually the whole process is approving the drug. So there are companies that specialize in this process, and they know exactly what to do, and these kind of companies, they did such a process for hundreds of times. So we already got a quote from several companies like this, and they can do it pretty quickly. So all we have to do is just give them the drug, and they can do the rest of the work.
So what do you call the structure? What do you call the structure that you attach the peptides to? This octopus that you mentioned, does it have a name? Is it a particular kind of proprietary structure?
Yeah, there could be the scaffold could be several things. So far, we use the PEG polyethylene glycol, which is widely used in the drug industry, and we use other things too that work. It doesn't have to be the same way. The most important thing is that you'll be able to attach many peptides on the scaffold. This is the most important thing.
Yeah, I guess it's like attaching.
The scaffold is not important, no?
Okay, it's like attaching tools to functional arms or tools to the scaffold, and they go in and do the work.
Yeah. The important thing here are the peptides, not the scaffold. The scaffold is just a scaffold. It doesn't do the work. The work is done by the peptides.
I understand. Yeah, I understand. Very good. So, what will happen in the next year? Are you ready to hand off one of these cocktails and these peptide groups to be tested?
So we already have the first mutator, and what we have to do now is just to pay a lot of money to this kind of company that would take it all through the rest of the clinical trials and bring us through all the regulations up to and get an approval to start the clinical trials. And in parallel, we're discovering more and more targeting peptides to more cancer targets. So we want to accelerate this process. So we intend to bring more scientists to work on it in parallel. And of course, we want to get as fast as we can to the clinical trials and start treating patients.
Oh, definitely. That's great. So what's the best way for people to find out more about what you're doing? And, should they go on the internet, or how should they find out more?
I didn't understand.
Yeah, what's the best? Do you have the website? Yeah, what's the best way for people to find out more and contact you?
Oh, they can contact me through the website. The website is www.AEBI-bio.com, and they can look me in LinkedIn. Many people approach me through LinkedIn. It's the most common way to approach me.
Well, very good. Well, Ilan, thank you for coming. I really appreciate it.
Okay, thank you.
Okay, take care.
Okay, take care. Bye-bye.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.